Welcome to the deep dive. So what if, everything we know about how to classify and treat a really deadly blood cancer is actually only half the picture?
Right. I mean, it's a terrifying thought, but a completely necessary one.
Yeah, because for decades now, our approach to acute myeloid leukemia, or AML, has been intensely focused on, well, the hardware.
Exactly, the genetics.
Right. We hunt for the broken bone, which in genetics is just a DNA mutation, and then we categorize the disease, estimate survival odds, and pick our highly toxic chemotherapies based almost entirely on that genetic code itself.
But the source material we're unpacking for you today kind of completely upends that paradigm.
It really does.
It forces this total reevaluation of how we even define a tumor. We're looking at a landmark paper published in Nature in July 2026.
And what was the title again?
It's called Chromatin Landscape in Epigenetic Heterogeneity of Acute Myeloid Leukemia. And the core thesis here is that if genetics is the hardware of the cell, clinical oncology has essentially been ignoring the software.
The epigenome.
Right, the epigenome.
Okay, let's ground this a bit for those of you listening who, track the genetic space. We all know you have roughly 2 meters of DNA just crammed into a microscopic cell nucleus.
Which is still wild to think about.
It is. And to make that fit, the DNA is tightly spooled around these proteins called histones, forming this really dense structure we call chromatin.
Right.
So if the genome is your master hardware blueprint, the epigenome, which is the chemical modifications, and the actual physical unwinding of that chromatin that acts as the operating system.
Exactly.
Yeah.
It dictates which specific genes are physically accessible to be read at any given moment. Imagine your DNA as this massive library of cookbooks.
Okay, I like that.
If genetics is the actual text printed on the pages, the epigenome is it's the series of bookmarks, sticky notes, and folded corners that determine which recipes actually get cooked today.
Wow. Yeah. And if the cell's operating system leaves the wrong genes accessible, it frankly doesn't matter if the underlying genetic blueprint is perfectly spelled right.
Exactly. You can have 0 mutations. But if the wrong book is propped open, the cell will still execute a cancerous program.
But the challenge has always been that you can't just run standard DNA sequencing to see which parts of the chromatin are unwound.
Right, because sequencing just reads the letters.
Yeah.
It doesn't tell you if the book was open or locked in a vault.
Which brings us to the methodology that makes this paper so staggering. To read this software, the researchers use something called ATAC-seq.
Yeah, ATAC-seq.
And rather than just, tossing out the acronym, let's look at the mechanism. It stands for Assay for Transposase Accessible Chromatin. How does this actually map the physical architecture of the genome?
So it relies on this engineered enzyme called a TN5 transposase.
Okay.
And you can think of this transposase as a relatively bulky molecular probe. It carries a specific genetic sequencing tag.
Right.
When you drop this enzyme into the cell nucleus, its sheer physical size means it just cannot wedge into the tightly coiled, closed-off regions of the genome.
The heterochromatin.
Exactly. It simply bounces off. It can only slip into the euchromatin, the open, relaxed regions where the DNA is actively being used.
So it finds an open pocket, and then what?
Once it finds that open pocket, it cuts the DNA and drops its sequencing tag right there.
Ah, okay. So it's essentially acting as a physical filter. It's kind of the biochemical equivalent of walking through a massive office building with a flashlight, right?
Yeah, that's a great way to picture it.
You're only mapping the rooms where the doors are already wide open. You sequence just those tagged fragments, and suddenly you have this high-resolution topographical map of exactly which genes the cancer cell is actively using.
And they apply this topographical mapping at an unprecedented scale.
How big are we talking?
Huge. They utilized what they call the ECHROMA cohort, which consists of 1,563 patients with a recent diagnosis of AML.
Wow, over 1,500.
Yet drawing from independent biobanks across Sweden and Japan. I mean, this represents the most comprehensive epigenomic data set for a single cancer type ever assembled.
That's incredible.
And when they clustered these 1,563 patients based entirely on their ATAC-seq profiles, so purely on which chromatin doors were open, they identified 16 distinct epigenetic subgroups.
And they labeled them A through P, right?
Exactly, A through P.
Okay, this is where I feel like we need to push back a bit on the existing clinical standard. Every oncologist treating AML right now uses the World Health Organization guidelines, or the ICC guidelines, which sort patients based on their genetic mutations.
Right, the hardware.
Yeah. So if we map these 16 new epigenetic subgroups against those existing genetic classifications, are the WHO guidelines just completely missing the mark?
Well, I wouldn't say they're wrong, But this data proves they are critically incomplete.
Okay, how so?
Because for a few subgroups, the hardware and software actually align beautifully. Take subgroups A, B, and C, for example.
What happens there?
These perfectly match known genetic fusions like PML-RRA. In those specific cases, the structural mutation is just so dominant that it forcibly dictates the epigenetic state.
So the hardware damage is so severe, it locks the software into a specific crash loop.
Exactly that. It's a hard crash.
Right.
But for the vast majority of the other subgroups, it's a highly complex web that doesn't match up neatly.
Right.
Standard classifications will group patients with NPM1 mutations or KMT2A rearrangements into distinct, separate genetic buckets.
Because they look different genetically.
Exactly. But the ATAC-seq data took these same patients and reorganized them into 4 entirely new epigenetic subgroups, D, E, F, and G.
Wait, meaning that structurally their DNA mutations were completely different, but their chromatin architecture was identical?
Yes. At the software level, their operating systems looked exactly the same.
That is wild. What were these specific subgroups doing that overrode their genetic differences?
They all shared a massive overexpression of HOX genes. The epigenetic software was driving the cancer through the exact same developmental pathway.
Wow.
Subgroups D, E, and F were packed with NPM1 and FLT3 ITD mutations, whereas subgroup G had almost none of those. It presented instead with KMT2A rearrangements.
So clinically, we've historically treated these as completely different diseases because the mutations are different.
Right. But at the software level, they are literally running the exact same leukemic program.
Okay, let's synthesize where we are about 5 minutes in, just to lock this in. So first, we're shifting away from treating DNA, the hardware, as the sole arbiter of disease.
Yep, the epigenome is the software.
Second, by deploying the TN5 transposase to physically tag that accessible chromatin across over 1,500 patients, this research reveals 16 distinct software profiles operating underneath the genetic mutations we usually look for.
Exactly.
It just raises this fascinating question for you, the listener, about how we categorize pathology. Think about the medical definitions we rely on daily. How often are we defining a disease by its structural damage while completely missing the active process that's actually driving it?
I mean, it's a profound blind spot in modern medicine. But mapping the open chromatin is really just the first step.
Right, because now you have the map, but you need to know how it works.
Exactly. The researchers then had to figure out how these specific open regions are actually driving the leukemia. And this brings us to the regulatory control room of the cell.
Which is governed by transcription factors and superenhancers.
Yes.
Okay, let's break those down. A transcription factor is basically a specialized protein that binds to specific DNA sequences to regulate the rate of transcription, right?
Yeah, you can think of it as a conductor standing at a podium, commanding a gene to turn on or off.
Right. But superenhancers, those take this to a totally different level. We know normal enhancers are regions of DNA that kind of loop over to boost gene expression. But superenhancers are these massive clusters of these regulatory elements.
They are, and their mechanism is honestly fascinating. Superenhancers don't just act as standard volume dials.
What do they do?
Because of their immense size and the sheer density of binding sites, they actually trigger a phase separation in the nucleus.
Phase separation, like oil and water.
Kind of. They create these localized high-concentration droplets of transcription machinery. They pull in massive amounts of transcription factors to aggressively and continuously drive the expression of cell identity genes.
So in the context of cancer, these superenhancers are basically the engines keeping the malignant state locked in.
Exactly. They are massive amplifiers hooked up directly to that conductor's podium.
And the paper found a staggering number of these engines, didn't they?
They did. Across the samples, they identified 1,718 non-redundant superenhancers.
That is a lot of engines.
It is. Now, about 500 were common across most subgroups, likely just keeping the basic cellular lights on. But almost 400 of these superenhancers were entirely unique to a single epigenetic subgroup.
Wait, really? Meaning subgroup D is powered by an entirely different set of molecular engines than, say, subgroup M?
Precisely. And to understand how these unique superenhancers were wiring the cell, the researchers deployed 2 advanced computational algorithms called Anansi and Coltron.
Okay, Anansi and Coltron. Honestly, those sound like secret DARPA projects. How do they actually model the biology here?
Well, they construct these mathematical gene regulatory networks.
Okay.
The Coltron algorithm specifically looks at the ATAC-seq peak intensity, which is basically how open a region is.
Right.
And it combines that with motif analysis, which identifies the specific sequence patterns where transcription factors bind.
So it's looking at the open doors and the specific keys that fit them?
Exactly. It integrates the superenhancer signal and the transcription factor binding data to calculate what they call an influence score.
Got it.
It builds this graph theory network to figure out which transcription factor is acting as the central hub, basically the dominant node controlling the entire network for that specific subgroup.
So it's not just a parts list, it's an organizational chart of the cancer's management structure.
That's a perfect way to put it.
So what did that management chart reveal about the different subgroups?
It showed us that the epigenetic software hijacks very specific, primitive differentiation pathways.
Can you give an example?
Sure. So the unique regulatory networks in subgroups D and O are dominated by transcription factors associated with hematopoietic stem cells.
The most primitive early-stage blood cells.
Right. But the networks in subgroups M and N are wired completely differently.
How so?
They're driven by factors like GATA1 and KLF1, which push the cell toward an erythroid lineage. They're mimicking red blood cells.
Wow, okay, that completely reframes the disease for me. The software isn't just universally commanding the cell to multiply uncontrollably.
No, not at all.
It's giving the leukemia a highly specific, subgroup-dependent developmental personality.
It goes even further than that. In subgroup K, the Cultron algorithm identified central hubs controlled by BCL11A and the IRF family.
And what do those usually do?
They normally regulate B-cell development. So the genetic hardware test simply reads as generic AML, but the software reveals this customized hijacked network of conductors and amplifiers mimicking entirely different branches of the blood family tree.
Unbelievable. Okay, taking a moment to consolidate this second milestone. We've seen that these super enhancers act as massive localized engines of transcription.
Right.
And they uniquely power central transcription factors depending on the subgroup.
Exactly.
By mapping these networks with algorithms like Cultron, we realize every single one of the 16 subgroups is running a distinct operational structure.
Yeah, a unique management chart.
It makes you wonder, if you apply this lens to other complex systems, how often are we trying to dismantle a network without actually knowing where the central hub is located?
All the time. You're hitting on the core limitation of blunt force therapies right there. But, any rigorous scientist looking at this bulk sequencing data would spot a critical methodological vulnerability.
Right. The blood soup problem.
Exactly.
Because bulk sequencing takes a bone marrow sample, which is just a chaotic mix of millions of cells. You have leukemia cells, but you also have healthy T cells, healthy erythroid cells, normal stem cells.
It's a total mix.
So if subgroup M shows an erythroid transcription profile, like you said, how do we mathematically prove that isn't just background noise from a sample that just happened to contain a high number of normal red blood cell precursors?
That is the million-dollar question, and it is the single most important validation step in the entire study.
How did they solve it?
To eliminate the background noise, they had to move away from bulk analysis entirely and zoom in to multiomics single-cell analysis.
Okay.
They performed simultaneous single-cell RNA sequencing and single-cell ATAC-seq on 281,167 individual cells.
Wait, almost 300,000 individual cells?
Yes, taken from 36 AML samples spanning the 16 subgroups alongside normal bone marrow controls.
I just want to pause on the sheer technical weight of that for a second. Analyzing both the RNA transcripts and the physical chromatin accessibility within the exact same individual cell scaled out to nearly 300,000 cells.
It's an astronomical amount of data.
Yeah. So when they resolved the data at that microscopic resolution, what happened to the background noise?
It completely separated out. It's beautiful. At the single cell level, the healthy control cells clustered in distinct normal developmental pathways, just as you'd expect.
In the leukemic cells?
The leukemic cells within a specific ATAC subgroup exhibited a universally shared epigenetic fingerprint.
Meaning what, exactly?
Meaning every single cancer cell extracted from a subgroup D patient had the subgroup D software installed, distinctly separated from the normal cells.
Okay, so it definitively proves the epigenetic profile is the intrinsic operating system of the cancer itself, not just some sampling artifact.
Exactly.
But single-cell analysis doesn't just give you a static snapshot, right? Doesn't it also give you temporal data?
Yes, and this is where it gets really interesting. They utilized what's known as pseudotime analysis.
Pseudotime?
Yeah. Pseudotime algorithms take the transcriptomic profiles of thousands of single cells and mathematically order them along a continuous manifold.
So it's essentially reconstructing the cell's developmental trajectory from an immature state to a mature state?
Right. It allows us to track a cell's lifespan even from a single snapshot in time.
Like mapping a human's biological changes from a toddler to a teenager to an adult, but by looking at their molecular exhaust.
Precisely. That's exactly it. And they applied this trajectory mapping to HOXA9.
Ah, that crucial transcription factor we discussed earlier for subgroups D through G.
Yep. In all of these HOX-related subgroups, HOXA9 is active throughout the leukemic cell's entire life.
Okay.
But the pseudotime analysis revealed that its activity peaks at completely different developmental stages depending on the subgroup.
Oh, wow. This is a really vital nuance. So it's not just that the gene is turned on, it's when the amplifier is pushed to maximum volume.
Exactly. The temporal dynamics dictate the pathology. In subgroup D, the HOXA9 activity peaks extremely early in the pseudotime trajectory.
Meaning what for the cell?
It causes a maturation block when the cell is effectively still a toddler.
Okay.
But in subgroup F, the exact same transcription factor peaks much later when the cell is closer to an adult state.
It's the same genetic driver, but shifting the execution timing alters the entire nature of the disease.
Exactly.
So as we hit our third mental checkpoint here, the multiomics single-cell analysis on nearly 300,000 cells doesn't just confirm the universal fingerprint of these subgroups.
Right.
It introduces time as a primary variable. The timing of when a regulatory network peaks is just as responsible for the cancer's behavior as the network's existence.
Yeah, it really is.
It's a striking reminder for all of us that analyzing what is happening is often completely useless if we don't map when it's happening.
And understanding the what and the when inevitably leads us to the clinical so what.
Yes, exactly. Because for a patient sitting in an oncology clinic tomorrow, does this massive computational mapping actually translate to better survival odds?
Well, let's look at how we predict risk right now.
Okay, the current gold standard for predicting AML risk is the ELN classification, right? The European Leukemia Net.
Exactly.
Oh.
And it stratifies patients into favorable, intermediate, or adverse risk based strictly on their genetic mutations.
So purely hardware.
Purely hardware. But does layering this new software map over the ELN hardware check actually change clinical decisions?
Does it?
Drastically. The researchers proved that integrating the 16 ATAC subgroups significantly improves prognostic accuracy over the ELN alone.
Give me a real-world scenario.
Okay. A patient might walk in and be classified as intermediate risk based on their DNA, meaning they might receive standard chemotherapy.
Right.
But if their chromatin map places them in a high-risk epigenetic subgroup, the oncologist now knows that standard chemo will likely fail.
Wow.
And they need to immediately pivot to a more aggressive intervention, like a stem cell transplant.
So better risk stratification is crucial, obviously. But what about the actual drugs? If we know how the gene regulatory networks are wired, did the researchers find a way to short-circuit them?
They did. They conducted extensive ex vivo drug screening against these subgroups.
Ex vivo meaning outside the body, in the lab.
Right, testing actual tumor cells with different drugs. And they uncovered highly specific vulnerabilities that the genetic hardware completely masked.
Can you give an example?
A perfect example involves MEK inhibitors and ABL inhibitors. Traditionally, you would only deploy a MEK inhibitor if you sequenced the DNA and found a structural mutation in the MEK or RAS signaling pathway.
Right. I mean, you don't use the targeted drug if the target's gene isn't mutated. That's Oncology 101.
Exactly. But the software mapping revealed that in certain subgroups, the epigenome is holding the chromatin around the MEK pathway genes wide open. It's constantly flooding the cell with MEK signaling. The genetic hardware of the MEK gene is perfectly normal. No mutations at all.
But the software is overdriving it.
Yes.
So the cancer is actually highly vulnerable to an MEK inhibitor, but standard genetic testing would have told the doctor not to bother using it.
Precisely. That is a massive paradigm shift for targeted therapies.
But I have to ask the practical question here.
Okay.
Doing ATAC-seq on living, pristine, frozen bone marrow cells is a logistical nightmare.
Oh, it really is.
It's highly technical, it's incredibly expensive, and standard community hospitals simply do not have the infrastructure to run it. If the test is too hard to deploy, how does this actually help patients at scale?
The researchers anticipated that exact bottleneck. They knew ATAC-seq was way too complex for everyday clinical diagnostics.
So what did they do?
They built a translational bridge using standard RNA sequencing.
RNA sequencing. But that's vastly cheaper and widely available in pathology labs today.
Exactly. It's highly stable and scalable.
But wait, RNA sequencing just reads the transcripts, right? It doesn't map the physical chromatin. How does that serve as a bridge?
Because the RNA transcripts are the direct downstream output of those specific super enhancer networks.
Oh, I see.
It's like looking at the financial exhaust of a corporation. You don't need access to the CEO's locked safe if you can reliably read the public quarterly earnings reports.
That is a great analogy. So how did they connect the two?
Using machine learning, they analyzed the massive ATAC-seq dataset alongside the RNA data. And they distilled the entire complexity down to a surrogate model of just 30 specific genes.
Wait, a 30-gene surrogate test? That's it?
Yes. By running a simple, cheap RNA expression test on just those 30 genes, the algorithm can accurately predict which of the high-risk ATAC subgroups a patient belongs to.
It just infers the chromatin state without ever having to run the complex ATAC-seq procedure.
Exactly. They took a mountain of multidimensional, phase-separated epigenomic data and compressed it into a clinically actionable, highly accessible diagnostic tool.
I mean, that is the hallmark of incredible science, right?
Yeah.
Mapping the entire universe just so you can tell the local clinic exactly which 30 stars to look at.
Truly.
Let's pull all this together. We began by questioning our fundamental reliance on genetic mutations, the broken hardware, as the absolute truth of cancer.
Right.
And through the lens of this monumental 2026 Nature paper, we've seen how techniques like ATAC-seq allow us to read the active epigenetic software.
We watched 1,563 patients get reorganized into 16 distinct subgroups,
completely independent of their DNA mutations. And we explored how super-enhancers and transcription factors build those customized regulatory networks.
Yeah, and how single-cell pseudotime analysis reveals the critical nature of developmental timing.
And finally, how all this complexity was elegantly distilled into a 30-gene RNA test that reveals hidden vulnerabilities to existing drugs.
It entirely changes the battlefield.
It really does.
But, it also leaves us with a really provocative final concept to consider.
What's that?
We're used to thinking of cancer as permanent hardware damage, a genetic mutation you can only deal with by poisoning or radiating the cell until it dies.
Right.
But if the cancer's identity is actually heavily driven by this epigenetic software, well, software isn't permanent.
It can theoretically be rewritten.
Exactly. As we map these chromatin landscapes, could the future of oncology shift away from trying to just nuke the tumor and move toward using targeted epigenetic therapies to fundamentally reprogram the software?
Literally coaxing a leukemia cell to simply mature back into a healthy blood cell.
Exactly.
Wow. It just makes you wonder how many unsolvable problems we face simply because we're trying to fix the hardware when we really should be rewriting the code.
Absolutely.
Well, thank you all for joining us on this deep dive. Keep questioning the structural truths of the world around you. And the next time you're handed a straightforward diagnosis, remind yourself, you might only be looking at half the picture.